The main mission of our facility is to provide a service that helps our users to answer their research questions. My name is Markus Hartl and I'm the head of the mass spectrometry facility at the Max Perutz labs. Our typical service process usually starts with me having a conversation with our users to understand the research question. I regard this a very important point because if we don't really understand what the user wants from us, we cannot deliver the correct service. Then we do first sample processing depending on which kind of service we're working on. Then we'll always perform quality control to assess whether the sample quality actually meets our criteria for further processing. Then we proceed to the liquid chromatography and mass spectrometry, the actual data acquisition to produce the raw data. After this, we do again quality control to assess whether the raw data meets our quality criteria. And then if everything is fine, we proceed to data analysis, where we actually analyze the data and try to make biological sense of it. And once this is done, we provide a report, and this report is then sent out to our customers. Usually, we offer to discuss the data and the results with our customers so that they can really understand what we deliver.
My name is Natascha Hadl, and my role in the team is I'm mostly focused on sample processing and all the organizational stuff around it, so ordering consumables and so on. When a customer brings their samples, I take them over and then I process them by, for example, doing a digest on the samples, checking them on our quality control system, and after that we prepare the samples, so we decide how much we want to put on the mass spectrometer and which method we want to use. We get samples that are on beads, so proteins coupled to beads. We also get in-gel samples, where the proteins were first run on an SDS page gel, or we get them in solution as a pellet. And depending on which type of sample, we also need to apply different protocols to the samples.
I'm Dorothea Anrater. I'm a staff scientist at the mass spec facility of the Max Perutz labs. My role is to carry out bigger projects. The quality control serves us to know how much of sample or protein we have. This reduces the risk of overloading the LC-MS system. It also tells us whether the digestion has been successful, and we also see whether there are contaminations. Then we would go for another cleanup. The analysis of a proteome is mainly peptide-based, and once we have these peptides, we can separate them with liquid chromatography. Separated peptides are then transferred to the mass spectrometer. In the mass spectrometer, they are manipulated by electric fields, and since they behave differently because of their mass, they can be separated. The instrumentation is focusing on relevant methods for peptide analysis. Proteomics field is rather young in the science, in life sciences. There's a lot effort in developing new instruments and making them more sensitive, which means that we identify more proteins with less material.
My name is Wolfgang Reiter. Besides the service that our facility provides, our group is also part of a research consortium. And I'm responsible for managing these projects and also to analyze all the samples that are provided from our collaboration partners. Our group provides a LC-MS platform to quantify and map acetylomes, phosphorylome and proteomes of the given samples. In addition, we do interactome studies, so we identify all interaction partners of a protein of interest.
My name is David Hollenstein, and my main task in the group is to develop and maintain our bioinformatics pipeline. We first measure the samples at the machines. This creates quite large raw data files, which then needs to be interpreted. For this we use either academic software or commercially available software. And then afterwards we have to apply post-processing to this data. That means we have to normalize the data, filter them according to certain quality criteria, and then perform statistical analysis.
I'm Wei Tian Chen. My main role in the team is to analyze the data, generate reports, and communicate the result with our customers. From the instrument, we get MS raw data. It's a big data file. So first, I open up the raw data and inspect it to see if it passes all our QC criteria, if everything is fine. Then I will check the submission form from our customer to understand the sample conditions and also what their aims are. Afterwards, I need to use specific software to search the data, to identify the proteins and quantify the protein in the data.
The turnaround times of our services certainly depend a little bit on the type of service. So, for example, for rather simple intact protein mass determinations, this can be a couple of days from submission to receiving the reports. For more advanced services, it can be up to 6 weeks. On average, I would say for our most common services, it's probably 2 to 3 weeks. Well, the unique selling point, in my opinion, is I think the interaction with our users. I think it's really important that we understand what the users need from us, that we understand the research questions. And because we are all in our team scientists at heart, we want to understand the science and contribute to that science. And I think this is really important in comparison to just submitting a sample, getting results, and not having much feedback on that. I think this is probably one of our most important added values that we can provide.